Evolution of Photosynthetic Pathways by Bob Ganesan. The earliest forms of life were anoxygenic, or did not need oxygen. Photosynthesis, as it is seen in plants, most likely originated from cyanobacteria that underwent endosymbiosis and horizontal gene transfer, and they eventually evolved into a rudimentary plastid. The aim was to gain energy in an environment that lacked oxygen. When these bacteria began to oxidize water as a source of electrons, oxygen was emitted into the environment. This caused oxygen concentrations in the atmosphere to rise. Photosynthesis can be divided into the light-dependent reactions and the Calvin cycle. This is how the light reactions evolved. At first, rudimentary organic molecules on the Earth were used as chemical energy by bacteria to make more complex molecules, and this can be seen in bacteria that live near hydrothermal vents and geysers. Over time, these molecules got depleted, so the bacteria that existed had to turn to alternate forms of energy production. One was with light. Bacteria evolved that could use the energy from light to make reduced compounds and high-energy intermediates that could be used in anabolic processes. In order to sustain this reducing power, these bacteria would use H2S. About 3.5 million years ago, bacteria turned to water for reducing power, causing the emission of oxygen into the atmosphere around 2.7 million years ago. Chlorophyll evolved as the light-absorbing pigment, and the energy trapped via electron transport was used to power the synthesis of ATP and the reduction of NADPH. There are many hypotheses that explain how these rudimentary photosynthetic bacteria with only one photosystem evolved into cyanobacteria that had two photosystems. One is that the bacteria evolved along two lineages, with one having bacteria of photosystems of type PS1 and the other with bacteria with photosystems of type 2. These two lines may have fused together via symbiosis until they became an integrated organism with both types of photosystems. Another is that gene transfer may have taken place without complete fusion of the two lines of evolution. A third possibility is that an early photosynthetic cyanobacterium that had a switch to select the gene to code for one photosystem or the other could have lost the switch, causing the evolution of a line of protocyanobacteria that had both kinds of photosystem. Within this line, another line of cyanobacteria may have evolved that was able to oxidize water as a source of reducing power. This performed oxygenic photosynthesis. This came about in the Archean eon. The earliest forms of life were carbon-based, and thus had to derive carbon from CO2 fixation. The earliest life forms layered various forms of carbon fixation mechanisms in order to obtain carbon, and this was controlled by many enzymes. One of these was rubisco. This fixates CO2 and O2. When it evolved, the environment wasn't oxygenated, so its inability to exclude O2 would have made little difference in this photosynthetic yield of early plants. So, by the Carboniferous period, all plants used rubisco for the carboxylation step of photosynthesis. This formed the C3 cycle. But when oxygenation occurred in hot environments, the fixation of O2 caused its breakdown into CO2 via breakdown of its two-carbon intermediate. And it did not also produce any ATP. So, it caused photosynthesis to be inhibited by over 30% at warmer temperatures. So, to prevent carbon wastage, C4 photosynthesis evolved, which involved the structural change of photosynthetic tissue. This was an adaptation that compensated for high rates of photorespiration and carbon deficiency. So, any environmental factor that enhanced photorespiration would select for traits leading to C4 photosynthesis. This included heat, drought, salinity, and low CO2. And these adaptations have been traced back to the ancestor of around 30 lineages of monocots and eudicots in the tropics. Another form of carbon fixation that evolved for the same purpose was the CAM pathway. Like the C4 pathway, it uses a dual carboxylation pathway, but it is not separated by a physical barrier, but by the time of day. CO2 fixation by PEP carboxylase occurs in the night when the stomata open, and CO2 fixation by rubisco occurs during the day when ATP and NADPH are made in the light reactions. This was traced down to a lineage in the plant family Crassulaceae. When cyanobacteria began using water as a source of reducing power 3.5 billion years ago, oxygen was released as a byproduct of this oxidation. When oxygenic photosynthesis in prokaryotes first came about in the Archean eon, the free O2 it produced probably dissolved in the surrounding water until it reached a high enough concentration to react with elements dissolved in the water. This caused the iron ions to react with the oxygen and form iron oxide. This released oxygen then dissolved into the seas and lakes, and eventually began to gas out of the water and enter the atmosphere. This caused the level of atmospheric O2 to increase at a gradual rate from 2.7 billion years to 2.4 billion years ago. It then increased rapidly to between 1 and 10% of its present level. This caused many repercussions. Oxygen attacks chemical bonds and can inhibit enzymes and damage cells. As a result, the rising concentration of atmospheric O2 probably doomed many prokaryote groups, causing them to go extinct. But those groups that were able to survive the spike in oxygen concentration were able to propagate the traits that enabled them to live. So, many adaptations evolved, including cellular respiration. The rise in oxygen led to the evolution of the first eukaryotes, which were organisms that have cells with membrane-bound organelles and nuclei and a well-developed cytoskeleton around 1.8 billion years ago in the Proterozoic eon. This is supported by the endosymbiont theory, that states that mitochondria and plastids were once prokaryotes that began living within larger cells. So, the hypothesis of serial endosymbiosis supposed that mitochondria evolved before plastids in a series of endosymbiotic events. Sufficient evidence supports this origin, such as the fact that the inner membranes have homologous enzymes and transport systems, the fact that they reproduce by processes similar to binary fission in prokaryotes, and the fact that their ribosomes are like prokaryotic ribosomes. After the formation of eukaryotic cells came the origin of multicellular organisms. This occurred in the Proterozoic eon. However, larger and more diverse eukaryotes do not appear in the fossil record until about 600 million years ago. This was radically different because before 1.8 billion years ago, Earth was only inhabited by unicellular prokaryotes and eukaryotes, and before the Ediacaran biota came about 600 million years ago, which are the larger and more diverse eukaryotes, the multicellular eukaryotes that were microscopic. So, the oxygenation of the atmosphere resulted in the eradication of some life forms, but led to an increased diversity of life that originated from those life forms that did live. Thank you.